Hello, biologists. Today we're going to talk about the epigenetics. But first, please look at who decided to hang out today. So if anything weird happens in my screen, it's because of him. Let's get started. We know that a mutation is a permanent change in the nucleotide sequence of DNA. Now, what if we want to change the way that a gene is expressed without mutating our DNA? In that case, we're going to talk about gene expression and epigenetics. The prefix epi means upon, so we're going to be making changes to the genome without mutating the genome itself. That's where the vocab word comes from. Epigenetics is really a process of controlling how a gene is expressed and when and where it's expressed. We have a couple different types of epigenetics. We have genetic modification, which is occurring in the genes, so on the DNA itself, using molecular signals. We have methyl markers and we have acetyl markers. If your gene is methylated, it decreases transcriptional activity. And if it's acetylated, it increases transcriptional activity. So the type of marker you add to your gene can regulate the gene expression. We also have histone modification. Remember, histones are the proteins in chromatin that DNA wraps around. Got a visual in the bottom right corner here. So we've got a histone protein here. It's got several different components, so it'd have a quaternary structure. And DNA wrapped around this collectively is referred to as chromatin. One chunk of it is called the nucleosome. And that chunk of DNA can slide back and forth around the histone. And so the histone can hide genes, or it can tighten up, or it can loosen up. If your DNA is loose and accessible, you can physically get to it and you can do transcription translation. If it's not physically accessible, it's hidden, it's wrapped around that protein. We call loose DNA euchromatin, U for true, and we call tight DNA that's condensed, that's not accessible, heterochromatin. We can actually see the difference between these types of DNA in the microscope. On the left, you can see this is heterochromatin. It's really condensed, it's super dark and visible. So these genes are tightly wrapped around the histone proteins, and they're physically not accessible for RNA polymerase II to transcribe. So I'm not going to be able to access that gene. I won't be able to make a protein from it. That gene expression is silenced. Over on the right, I have these cells. I can see the nucleus, but they're paler. They're less filled in. That's because the DNA is loose. It's not wrapped tightly. We call that euchromatin. There is space for the cellular machinery to access that DNA for transcription to occur. Here's another visual. The blue is the histone proteins. Here's the gene that we would be interested in. It's tightly packaged up. You can't really get to it. I've added some methyl groups because that decreases transcription. That means that this gene is effectively silenced. If I wanted to access that gene, I would loosen my chromatin. It would become euchromatin. I could put some acetyl groups on instead of methyl groups, which increases transcription. And now I can activate this gene and use it to make a protein. When we change whether or not a gene is being used, we have three basic levels of doing it. One is transcriptional. So we're going to modify what genes are available with chromatin remodeling. That's those euchromatin and heterochromatin, acetylation and methylation. And then we also have transcriptional regulation, where we are identifying what part are we turning the information into mRNA. And then after we create the mRNA, how do we process it? That's RNA processing. That would be which mRNA introns we're removing. Do we change the order of the exons? How are we modifying it afterwards before it goes to the ribosome? In translational gene expression control, you would identify whether or not your mRNA is long-lived or short-lived. If it's short-lived, then you won't be able to create very many proteins from it because it'll break down quickly. But if it's long-lived, you might be able to make a lot of protein from it because it's around for your ribosomes to work on. That would also change the expression of the gene. So degradation is an important thing, the lifespan of mRNA. And then as it's going, we can make little changes to the growing polypeptide with the tRNA as we're translating it itself. We'll talk about that in just a second. And then the last way that we regulate the genes is by changing the protein after the protein already exists. That's post-translational. In post-translational regulation, you could fold it differently, you could add different chemical markers, maybe you make it short-lived so it only works for a period of time, or you could ship it off to different parts of the cell where it'll have different uses. All of that is ways to control how the gene is being expressed without modifying the gene itself. As a fun fact, prokaryotes do transcription and translation at the same time. And so they usually do all their modification transcriptionally because it happens together. Let's talk a little bit about how we can do translational modification. One of the ways is with wobble pairing. A wobble pairing is a tRNA that's anti-codon region is normally perfectly bound to the codon region of the mRNA. And in wobble pairing, you'll have a tRNA that doesn't have an exactly appropriate anti-codon region, but it binds close enough that it slips in and it's used. So we're essentially creating a missense mutation without mutating the DNA because we're altering the tRNA that's binding to it. This can also be controlled by promoting or blocking the accessibility to the mRNA. We also have small ribosomal RNAs, sRNAs, which are really interesting because they're new. We're doing current research on this and finding out that these sRNAs may be involved in regulating mRNA activity and ribosome activity way more than we expected. So if you find this entire process fascinating, feel free to do a research dive on that. It's really interesting. I don't expect you to identify anything in here for the exams, but it's cool to know that the absence or the presence of these sRNAs will promote or decrease translation. So there are other controls that we don't fully understand. In post-translational gene regulation, the way that we fold and how stable the protein is affects its longevity and its function. We also can decrease the active level using chemical alterations or physical alterations like folding it differently. And then how long your protein is in existence affects what it's going to be doing. And we can also change our alterations after we add them. So one protein can have many different manifestations. And that brings us to a new word in vocab, the proteome. In the proteome, you're thinking about the entire complement of proteins that can be expressed by a cell. It works like this. We expect our human genome to have 20 to 25,000 different genes. In theory, each gene codes for one protein. In reality, because we do different types of gene expression control transcriptionally and translationally, each one of those genes actually codes for about 100,000 different types of mRNA. And then those mRNAs can create over 1 million different types of proteins. Because of wobble pairing, because of decreased or increased activity of the ribosome, of the different folding, different chemical tags. So our idea that one gene equals one protein isn't really true. One gene can equal many different proteins because we're expressing how it's controlled differently as we go through the cell. There's implications of this as well. We're finding out that the epigenome can be inherited, which means that the genetic changes that occurred to your parents can end up in you. And what's really wild is that it can affect more than one generation at once. So if you are a male, you're constantly producing sperm. If there is an event that happens and you get an epigenetic marker in a cell that's going to become sperm, and that sperm fertilizes an egg, you've then passed on that epigenetic marker to the next generation. So men are capable of affecting A, themselves, and B, their children. But here's the really wild thing. Imagine that that child is a female. All women produce their eggs in utero. So you are born with all your eggs by the time you are birthed. And so if this person is a female and they have the epigenomes of both their parents, they're going to put those into their eggs by the time they are born, and their children will be affected by their grandparents. So it's really a wonderful, complex system. We're finding out now that environmental effects and trauma can be inherited epigenetically. There's a lot of really interesting current research happening about this. The first thing I want you to do is watch this mini news report about the agouti mice experiment. And after you watch that, I have 3 articles for you to read. We have reviews of current articles coming out. So each one of these is a review, not the article itself. It's going to tell you what the study found. So that's why it's from yale.edu, hopkinsmedicine.org, livescience.com. So these are not the research articles themselves, they're reviews. And it's talking about the effect of epigenetics and how it's changing our understanding of inheritance. The first one is violent experiences alter the genome in ways that persist for generations. Super interesting. Scientists identify a new epigenetic approach to target colorectal cancer. And then epigenetic memory may help explain why PCOS tends to run in families. Check out each of those articles and then watch this review animation of the process of transcription, translation, and protein production. And then when you're done, I have a worksheet for you. The worksheet has some blanked out images just like this, and then I have a list of vocab on the left. I want you to find the place where each of these vocab words should go. Some of them are used more than once, and figure out where they're missing on your diagram. Once you're done, I want you to verbalize it out loud. So like tell your cat, tell your best friend, but walk through the process verbally, and then move on to the next one. You have a second diagram here of DNA replication. When you're ready, I've got the answers for the diagrams in this video. So you can pause it on this or pause it on this, but I want you to try and find all the locations first. And then lastly, I have a worksheet to help you identify which steps occur in which process. So here we have a chart. We have an option for DNA replication, an option for transcription, and an option for translation. So I'm going to check whether or not each item occurs in one or all 3, or maybe none of these steps. The first item is requires primers. I require primers for DNA replication, but do I require it for transcription translation? No. We're going to use transcription factors, and then we don't need a primer for translation. We use the start codon. What about makes a copy of a specific gene? Not replication, that's the entire genome. Yes, transcription, we're making a copy of one gene into mRNA, but not translation because we're using the mRNA to make proteins. So you're going to go through the whole table and check yes to which one of these 3 processes it is. And some of them will have more than one checked. So it begins with a start codon. That's true for transcription and for translation, but it's not true for DNA replication. Awesome. I hope that helps. Email me anytime with questions, and you have a good day.